Hello, everyone. My name is Gila Kahila Bargal. I would like to thank the organizers for giving me the opportunity to present my talk virtually, and I do apologize to you all for not being there in person. The golden jackal as a vector for zoonotic diseases was the PhD thesis of my student Ro'i Lapid, co-supervised with Kobi Marom Gilad. And I would like to thank them both, including my collaborators, that we had a very a great journey together. The golden jackal is a medium carnivore of the Canidae family. It is widespread from the east, from Thailand to the central west of Europe. In Israel, it is mainly along the Mediterranean region. In the last few years, the golden jackal has expanded its distribution, going into Ukraine, Poland, and Germany, and in Israel, going into the arid areas. This was because the golden jackal has adapted to a human, and it's actually a synanthropic species. That means it's a species that benefits from living in proximity to humans, yet remains beyond our control. As an omnivore, a synanthropic species with high distribution and high reproductive rate, the golden jackal has the potential to be a reservoir for many pathogens, especially zoonotic disease agents. In Israel, in Tel Aviv, you could see the proximity of the golden jackals to humans and their pets. So the interaction between the wildlife, the livestock, the domestic animal, and the humans causes more than 60% of human infectious diseases. And over 75% of the emergent diseases are zoonotic in origin. So the One Health approach emphasizes the health of the people, the health of the animals, both wild and domestic, and the health of the environment, all that are interconnected. So early detection of emerging diseases has an important role in implementation of preventive measures. In Israel, in the case of the rabies disease, we could see three different agencies that are taking place. Israel Nature and Park Authority are responsible for oral vaccinations of the wildlife. The Ministry of Agriculture for vaccination of domestic animals and dogs. And the Ministry of Health is responsible for the human health. So our research goals were to determine the role of the golden jackals as a emerging infectious diseases reservoir. Both the host itself, the pathogens that he is carrying, and their interactions with the environment and humans, as we could study through the microbiome and the resistant genes that the jackals are carrying. We believe that our results will contribute to conservation medicine, recommending strategies for management of the golden jackal population in order to prevent transmission of zoonotic diseases and enhance public health. The cohort consisted of 111 carcasses, all collected by the Israel Nature and Park Authority from four different regions in Israel. It is important to say that the carcasses were not killed for this research. The four different regions varied in their habitat, in the flora, and in the climate conditions. Region number 4 at the north of Israel was the coldest one with the highest average of precipitation, where region number 1 along the Jordan Valley was the hottest one with the lowest precipitations. Region 1, 3, and 4 were hotspots for rabies, while region number 2 along the shore was the control. Except of these differences between the different locations, the sanitation conditions also varied due to the different practices of the farmers. At the north of Israel, region number 4, most of the farmers grew chicken, where in region number 1 it was fisheries, and in region number 3 it was roaming cattle. The control region had different practices. Each practice brings its sanitation condition that can influence the animals. The cohort was equally divided between males and females, and most of the carcasses were adults or sub-adults with very few juveniles. The physical conditions of the jackals was tested, and we could see that over 92% of the carcasses showed normal body conditions. Only 7% were thin, and some of them had also external parasites. In order to achieve our goals, we sampled different types of samples, from fecal to fair to serum. Each one of them went through different analysis, and the results of all of them were together collected and compared and associated to each other. The genetic profile of the population was interested, as each individual had its own unique profile using 17 STRs loci. Yet, no association with the location of sampling was found. That means there was low genetic differentiation between the subpopulation and a gene flow between them. We also sequenced the three LTR's genes, LTR2, 4, and 7. Those LTRs have key roles in the innate immune system. The sequences show the same phenomena with no association to the location of sampling. The importance of this data is because the TLR activity can affect the gut microbiome through pathways of immunity and metabolism, and we wanted to associate it to our results. Testing for the pathogens that we could find, the first thing was to look for rabies. None of the individuals were rabid, but over 50% of them were exposed to the oral vaccination. Two zoonotic diseases were detected in high percentage, Neospora and Toxoplasma. The importance of these two pathogens is that they can cause serious diseases among domestic animals and humans. As sentinel species, rabies can detect the risk of resistance bacteria in the environment, which is important for both humans and animals. Among the 24 antibiotic groups, we could see two antibiotics that were abundant in the sampling, the tetracycline and the microlide. Checking, we can see that both of them are widely used in the agriculture industry and also by the veterinary medicine. So we decided to sequence 5 antibiotic resistant genes, and we found that for three of them, most of the jackals were resistant. Other association to other traits didn't show such significant role, except of slight significance to the geographical sampling location. The most interesting thing is among the positive tested jackals for toxoplasma, 25% of them were resistant to one gene, the beta SHV. The metagenomic analysis showed the abundance of antibacterial resistance genes and found 431 genes. The profile was similar among all 17 individuals that were tested. The most important thing is the theta A gene that clinically is responsible for resistance to tetracycline. So then we studied the microbiome, which actually let us put together all the data. From previous studies of microbiome, we know that the microbiome has an influence on the digestion of food, the creation of vitamins, and the function of the immune system, and also in some behavioral aspect. Furthermore, there is an axis between the gut and the skin through the immunology and metabolite pathways, and that's important in our case. So the bacterial phyla that were abundant among the two microbiome that was tested showed that in each one of them there was no special association to a geographical region. All shared almost the same profile of abundant bacteria phyla. The ones that were unique and differed stayed in the role of the others. The important thing is the different abundance of the phyla between the fecal samples and the FAIR, and we could see that each one of them had a abundant phyla that differs from the others. So when we check the species richness among the two microbiome, we could see that the FAIR microbiome was richer with significant differences associated to the regions that they were collected, while the fecal microbiome had less variety, with only a significant difference between region number 3 and number 4. Two other associations that were significant are the exposure to the oral vaccination and those individuals that were positive to canine distemper. Overall, we could see that the abundance of bacteria in each of the microbiome differed, and the FAIR and the fecal had some common bacteria, but the FAIR had more variety in its profile. The association of the microbiome to the age of the samples, it's very clear that the fecal microbiome, all ages share most of the bacteria, 90% of them. Where with the FAIR microbiome, only the subadults and the adults share the majority of the bacteria, 66.6%. Meaning that actually for the FAIR microbiome composition, there's a strong influence of the environment from the different location that it was sampled. Comparing the fecal microbiome to the host genetics, we could see that the closest genetically the samples are, also their microbiome profile or composition is, both for the fecal microbiome and the FAIR microbiome, indicating that there is an influence of the host genetics on the microbiome composition. So to conclude, I think we gathered a novel data on the golden jackal microbiome, antibacterial resistance, and its traits. And also we improve our understanding on the interface between humans and the wildlife, especially when we have cases of high prevalence of pathogens that are zoonotic in origin and can cause damage. For us, for the rabies interface, the data can help with targeting the oral vaccination and preventing the spread of rabies in Israel. An overall better understanding of how the human waste, especially antibiotics in the water and other products, can influence the pathogens and the antibacterial, antimicrobial resistance carriers. We believe that we show that the golden jackal can serve as a good indicator for anthropogenic impacts on the environment. I would like to thank all the people that help this research take over and for you for listening.